Certificates you will receive after the completion of the course, maybe next day or day by next. So that's not a problem. Mhm. Uh, in blotting techniques, we will go through each by each all those techniques that we had studied. Mainly, my focus will be on Western blotting technique, which is quite highly used in the labs. So I will show you. Yeah, I will try to have virtual lab also for that, and teach you the principle behind each part of it, like why we use nitrocellulose membrane, what is the reason behind using antibodies, primary antibody, secondary antibody, and yeah, beta means please thank. It's just say it's a beta shown means just to say you're welcome. Review article due date is 3 days after your or 2 days after your course is finished. So if it is finished on 3rd of September, so we will not take off on Saturdays and Sunday. We will keep continuing it. Yeah, we will keep continuing your sessions, and yeah, now after the sessions are over, after 10 days, you can have 2 or 3 days to do so. Yes, there is a national level exam, C2 level, and TEF is for France. I forgot the name. There is a national level to clear that. Any country, your PI should be good. Even in India, it's also good. Our Don't forget that our Indian institutes are any lower. Many of my friends they have published really good journals in really good impact fact paper while studying in our Indian institutes. So don't judge a book by cover. So I'm saying a lot about Germany because I had that experience, but I have to say our India is also not far behind. Our many people who did their PhD from here, they also went abroad afterwards. It's just because of the quality of work that we are doing is really good. India is not far behind anymore. We are in very quite good in numbers. So many of our friends now for the USA H1B visa is also open, so you can also go for postdoc or any PhD level studies there. So USA is again open for Indian roots, and let the people know that what the level of education system that we had is still very strong and quite what the words I want to say highly intellectual, because we study a lot of things in our school times, and the foreign peoples they don't do so much studies in science, maths, uh, many other subjects. A week would be too much. A week would be too much. So some lagging is happening today for some strange reason. Okay, we are back. We are back. If you start today, so today is 28th. I hope you have already started the planning for it. So time is not less. You can do it within 1 week also. So try to think every day. Put at least 6 to 7 hours to it, like full time to it, and then go for it. Oh, danke schön. So it's 8 o'clock. I'm quite flattered. Uh, but there is a lagging is happening for some strange reason. So I hope. Uh, No, after the sessions are over, you can send it within 3 days, so it will be fine for us. Aha, this is happening because of this. So I will send them later. So my internet speed becomes slow because of these video sendings to you. I guess so. So this lagging will not happen anymore, I guess. Okay? Okay, it's 8 o'clock. Let's wait 5 minutes more. Let more people join us. CSIR Net of Biotechnology. That's Bache, I'm about to start that for teaching GATE and CSIR Net actually. I haven't seen the syllabus yet, but I will also like to clear that by myself, that exam, because if I want to teach someone, so I should clear that exam also. Because for my studies, everything was automatically done. So I cannot say things which I am of not uh, aware of. But once I know things, then I can only speak about that. Okay? Don't worry. The more you extend your dates, the more will be you will have your certificates late then. So that's why I'm asking to do it ASAP. So it should not be very big because if you put some quality time to your writing your review, uh, yeah, yeah. So when you're writing your thesis, your master thesis, you put a lot of hours to it. You sit in the morning, like 8 o'clock, and you continue till 12, have a break of 30 minutes, eat something, then again start, and finish by 4 o'clock, have a break again, then start at till 7. So within that day, you can achieve a lot of things. So don't have excuses are bad. Don't take excuses. Excuses are for the losers. Yes. And you are not losers. You are all winners. You have to achieve heights of your success, of your dreams, wherever you want to go for. Yeah. Yeah, I understand 9 is dinner time, but most of the people, 8 to 10 is the most quiet time for every students here, and we can join it. Okay, it's almost 20:05. I will start now. Uh, with the cloning. So this was some things that we did yesterday. Yeah, we have to start from here. Okay. So are you all ready for the next class today? Yes, we're gonna start the session now. So all good? Everyone is up? Good. So basically, cloning, you might have studied that in your bachelor's or in your master's also. That it's basically we bring out our an external external gene with gene of interest to the plasmid, and then with the help of cloning we generate multiple copies of it in the bacteria, and then we extract the gene of our interest via breaking the walls of that bacteria and get the things that we are looking for. That's basically the basic principle of cloning is. So let's go in detail about it now further. If you want to do DNA cloning with plasmid vectors, what do you need? First of all, you need a plasmid vector. A plasmid vector should have uh, origin of replication. That's the most important, from where your reaction will start. Then there is a some ampicillin resistance genes should be there, and then third one is the multiple cloning sites where your various restriction enzymes could attach, and cut the plasmid and let the enter your gene of interest to be attached here. So this is the reason into which exogenous DNA can be inserted, and these are various polylinkers: HIN3, SPH1, PSTL, SAL1, XBL, BAMH1, HFAS, SMAL, KPN, SAC, ECORI1. So these are the various restriction enzymes. So recombinant DNA technology enables to produce large numbers of identical DNA molecules, and that is known as DNA cloning. And cloning can be typically generated by placing a DNA fragments of interest into vector DNA molecule, which can replicate in our bacterial host cell. So whenever a single bacteria or single vector containing single DNA is introduced into host cells, large number of fragments are reproduced along with the vector. So two common vectors that are E. coli plasmid vectors and bacteriophage vectors. Bacteriophage lambda vectors. So here you can see how in general the DNA cloning is done. On the left is the plasmid vector, and on the right you can see a DNA fragment to be cloned. Okay, then we with the help of enzymes insert this DNA fragment into this recombinant plasmid. It's being inserted with this green color. Then further we mix with the E. coli cells here this plasmid in presence of calcium chloride and culture in the nutrient agar plates containing ampicillin. So the bacterial chromosomes will have some these. This is the bacterial chromosome of this E. coli, and some of those one has been transformed. This is called as transformation. Whenever they take the plasmid out of it, or they have taken the plasmid inside, and there will be some cells that will not take plasmid and they die because of on the ampicillin plate. So this is an ampicillin resistance gene is also being added. So we can be sure that the cells that are surviving, they have that gene against ampicillin, and that's why they are able to grow. And we see them with the different colors then. Then independent plasmid applications will happen like this. Then cell will multiply like in this huge amount. Within each cell, these plasmids will multiply, and then cloning of cells of each containing copies of the same recombinant plasmids will happen. So that's the basic principle. Wherever you have a diagram to write in subjective exam or you have a question in objective, you can remember this diagram. Very simple and easy to understand. No gimmicks or any big diagrams to understand it. So there is also another way you can do multiple copies of DNA fragments actually, with different characteristics to that, and you can grow them also. So like in this case you have this our plasmid vector and various different DNA fragments of different functions. So like say we have four ones. So we again with the help of enzyme inserted them into the plasmid, and we have our these plasmids ready, four plasmids, and then transform into E. coli cells and select for the ampicillin resistance colonies, and each colony presents one clone. So like they will grow separately from each other because their function and their likelihood they like to grow on their own, so that's why they separate from each other. So this is a group of organisms with identical DNA, so four groups. Then how you cut these our DNA? It's we cut it with the help of restriction enzymes. So they produce two ends, sticky ends and blunt ends. Sticky ends are the one which can helps to make you uh, a way to addition of external gene to be there in an easy way. Whereas in blunt end it's quite difficult. So in sticky ends, it's also called as palindromes. Why is it so? Palindromes. Palindrome means like Anna. If we speak Anna from the left, it's Anna. If we speak from the right, it's also Anna. Auto, radar, kayak. There are many others name also like that. So whenever we speak from left or right, they will both mean the same name. So that's why the sticky ends produce these same way. Like here, if this E. coli when it's come E. coli 1, E. coli 1, when it comes, what it will do, It will cut from here and other from there. So you have both CTTAGA and CTTA. So same name are cutted like that. So like this, you have these sticky ends like ATTT, AATTT. Okay, so in restriction enzymes are site-specific DNAs generating double-stranded breaks. Until now, 600 naturally occurring restriction enzymes have been identified, and they recognize 4 to 6 base pair sequence patterns. Restriction cuts are more often asymmetric, generating single-stranded overhangs, and restriction cuts are also frequently symmetric. Whenever it's overhangs, then it's a sticky ends. When it is symmetric, then it's a blunt end. So these are the various examples of enzymes, restriction enzymes, and the source of microorganisms respectively, and how they cut at the recognition sites. This comes in exams actually, in many objective type exams like BAM H1. It cuts from GAATCC and it produces sticky ends like all. So there is a SMAI. This is come from the Cerateria marcescens. So it's cutting in the middle of here. CCC, GGG, GGG, CCC. So it's cutting in the middle somewhere. So it's producing a blunt end in that way. So remember these hint 3, Ecori I, SOW 3I, BAM HI, not L. So all these are very important. Mm, Now, sometimes restriction fragments with complementary sticky ends are ligated easily. So let's say we have this DNA1, DNA2, but in DNA2 we have three different types like AATT, AGCT, but somehow it will attach to the complementary end base pairs to this one, to only A part. So rest will remain unfragmented like B and C. And then with the addition of ATP and DNA ligase, we join them together and you have again a recombinant DNA. So this is also uh, a new way to make a recombinant DNA. So a small video about it now.
And are therefore able to replicate independently within a cell. Plasmids can be
Just wait a second.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white.
Okay. So that was quite self-explanatory, like what we studied before with the beta galactase example and other way why cloning, how it works. So let's continue. There's another method here for you, uh, like so sequence. We have a sequence of polylinkers where your all restriction enzymes could restrict actually. Okay, uh, Parmachite, LPN, Expal, Pistia, Hind, everyone. So we insert this E. coli restriction fragments to it. Okay, with the sequence of polylinker, our E. coli restriction fragments has been added. So this is your plasma vector with ampicillin resistance gene ORI and this polylinker to it, and our genomic DNA, that's external. And we cut them both with the help of E. coli 1. Yeah. And then one of the part with the help of DNA ligase and ATP, it's got attached to it. This is also one way of your one type of cloning that's happening. So, cloning the transcriptome. Now, if you have transcriptome is the one which includes your all mRNA and cRNA, mRNA, your ribosomal RNA, tRNA, and you only want to extract purified mRNA. In order to do so, what you do is you produce a complementary DNA libraries that are prepared from the isolated mRNA. So you have a oligo DTT matrix on which your T thymine has been attached in a 4 chains, and you have your mRNA with the 4 to 5 of adenine base pairs, and you also have the tRNA and also ribosomal RNA. So this is a mixture of our cytoplasmic RNA. So we mix them together, we hybridize them together, and because of hybridization, this mRNA will attach with different A amino acids and attach with a TTTT like this. And then we wash away the ribosomal RNA and tRNA and elute column in this low salt buffer, and hence you have at then your purified mRNA like this. And how to prepare your lambda complementary DNA library? Your lambda complementary DNA library, how you can prepare that? It's sorry, just some disturbance here. So you can see here in this diagram, but it's I think small for you to list. Can you see this? So we have 3 prime poly A tel here. Yeah, we hybridize with the DT primers, oligo DT primers in the first step, and then transcribe this RNA into complementary DNA, and then like here. So from this addition of this our oligo DT primer, we had our complementary DNA, and then we remove this RNA and alkali added poly DG tail. So we remove the tail also from the previous one, and we add this poly DG tail. So we have GGGGGG here and TTTTT here, and then oligo DC primers are added then on top of it. So here C primers are added, and then we again synthesize our complementary strand of our cDNA. So like this. So what we did, we had our first mRNA, and then from mRNA we added our oligo DT primers here like that, and then we generate complementary DNA out of it. Then we remove the RNA and and with alkali, and we added poly G tail here. Then on top we added DC primers, yeah, and we again generated a complementary form there like this. So then again, what happened then? Uh, I think just wait a second. I think you people might need it to be Zoom, I feel like. So I just, what I said, uh, I hope you can understand now. So this one, second step, transcribe RNA into cDNA, then remove the RNA, you have a complementary DNA, then you G tail, TT tail, then C is there, then C produce again complementary, then AATT is there. So these 5 steps are done. Then so we protect this complementary DNA via methylation. We add methyl group on both sides on top onto our double stranded cDNA, and then we add our E. coli linkers to be added on the both sides of it, like here GATTC, GATTC, and then we cleave with the E. coli. And then because E. coli cleave here at the A and here, so we cleave it. So here we have, now in this case we can ligate to the lambda arms package into the infected E. coli. So individual lambda complementary clones are being made here for you, which could be used for various applications. Clear now? So these are like, we are getting really more into complex cloning part, because it's good to know various other things what's happening within this field also. Then how to identify, analyze, sequence clone DNA. You can see it here that the most common approach to the most common approach to identify specifying clones involves screening a library or hybridization with radioactively labeled DNA or RNA probes. So labeled nucleotide probes are also used to identify a specific nucleotide sequence in a complex mixture of DNA or mRNA. Further, direct sequencing is also often applied in this case. So single nucleotide sequencing or large scale sequencing could be done. So this, let us come to the point of this DNA sequence, the Sanger dideoxy method, which is quite old method, but still people are using it because of its application. But it has been overtaken by the big market of next generation sequencing, like Illumina, ion exchange, ion torrent method, ligation method. So there are many companies are available who could do your sequencing of a human or any other model, mouse model or Arabidopsis or any bacteria. They can let you know within few days, like in 3 days they can do for 15 people this genomic DNA. So it's so fast now, because earlier it took around 23 years to do for the one person only, this genomic library of one person. So this technique of DNA sequencing, Sanger deoxy method, it's work on adding various DNTPs actually. So what you do, I mean, it's I made this presentation is good, I understand, but it will be more interesting if we play this video, then come back to that part. Yeah. So I will stop, because these all techniques, they are easy to understand. Sorry, just a second. I will play the video. Hello. Okay, so I will take care of that. ठीक है सर, ठीक है सर. Okay, sir. Okay, sir. Okay, sir. Okay. So let's continue with the video part. So mainly what we can see that what you have seen is that you have a primer, you have a template, yeah, whatever you're looking for. Then you add some DNA polymerase to that with the help of DNTPs with 100 micromolar of concentration. Then you add separately DATPs, DDDTPs, DDTTPs, DDCTPs, different forms. So your template DNA here, it will stop at some point and give a fluorescence out of it. Yeah. Like in this case, it goes like this till here, it goes like till here, and then it's keep going. And same way for G, same way for T, and same way for C. And it's gave different fluorescence colors while doing so. So denature and separation of daughter strands by electrophoresis. Then we can check these denaturation and separation by electrophoresis. So here you can see in the electrophoresis gel, um, you can see GGGCAT. So then like we put them in the gel, all these four different samples like we show you in the video, and then we arrange them in like this, and with the help of radiogram we see the graphs like green color, red color. Green is for the C is coming, then A is coming, uh, then some mixture of both are coming, the black one, then the blue is T is coming, then again higher peaks are coming. So like this we give the name A T T G G C T T T A T T C G, and this way we can have our whole genomic DNA sequencing. Okay, we have our whole genomic DNA sequencing like this. So that was Sanger deoxy methods. But now in the present, in this world, we have this next generation sequencing, which what in like in 2004 in this diagram you can see published in the Trends in Genetics that around the number of base pairs that it took them around 40 base pairs 1 month with the help of next generation sequencing. They took it 1 month to generate that data, but now with the time you can do it within in couple of hours actually to generate the multiple sequencing of human being with the help of various technologies such as Roche 454 FLX, Illumina, Solexa, Applied Biosystems, Helicoscope, Pacific Biosciences, Life Technologies, and Torrent. So they are quite old now, the names are. But now in 2019, these were made like in 2019. It's really fast. So there is a Mahajan Imaging in Delhi. They are using the latest next generation sequencing to analyze the COVID patients actually. So they already started to do with the COVID patients to check their genomic DNA, what happened to their genomic DNA, and they already started to accumulate so much data so that which could be used for our future scientists. So they are giving that data to the all pharmaceutical companies who are working on the working for the our vaccine, so that it could be helpful for us. So they say AstraZeneca from Oxford, I guess they want to do some trials in India now, but let's see how we will be successful. But it most likely is like 2020, the COVID time we are having. It seems um, that it's hard to find vaccine in this year, to find anything to reverse it. But as much as the movement started like in Delhi here, no one cares about COVID anymore. So this way the rate of transmission uh, might be rate of transmission might be reduced the more we are moving. Might be it might be reciprocal to each other. So I hope things get better for all of us and we can have a same life like we had before. The offline classes, we could go to universities, have a good time with the friends, play some soccer in the evening, and have a back same life again. So over to that, let's continue. So what are the parameters that we check in the next generation sequencing? Are the cost, read length, speed, accuracy, preparation time, manipulation steps. So these are the various steps that we take in that. So this is diagram you can go for to check the various pros and cons about various techniques that we have available from the Roche, Illumina, APG Solid 3, Pollinator, Helicos Bioscience, Pacific Bioscience. So from various companies, these are the various next generation sequencing platforms that are available. With their what is their chemistry behind is what are the libraries that they are taking, how much read length basis they are doing, how many days they are taking it actually. So you can go through this. Um, so this is a short summary out of it with the Roche, Illumina, and Solid, three different platform working on different principle. That is the chemistry behind the first one is pyrosequencing, second one is the polymerase based, third one is the ligation based. So the amplification that is done in the first case of our DNA is by the emulsion PCR, and the second one is with the bridge amplification, and third one is also again the emulsion PCR. So how many base pairs do we paired? It's 3 kilo base pairs, and in the second is 200 base pairs. Then in third is a 3 kilo base pairs. Uh, then how many mega mega base pairs that are run? These are the paired ends and the mega base pairs. 100 MB, 1300 MB, 3000 MB. So time per run 7 hours, 4 days, 5 days. So Roche seems quite good in 7 hours. Read length 250, but this is quite hot actually. Roche is quite hot. So 250 base pairs, 32, 35. Then cost per run is 8,000 in this case, 8,400, 8,900, and it's the most expensive, 17,400. Yeah. But per MB it's cost around 84, 5.97, then 5.81. That's how the rates are. So about these are the various diagrams for the pyrosequencing for the Roche, the principle behind it. So in this case, you first have your DNA, then you library is constructed in this way. They are unpaired, then put into our Appendorf, then emulsion PCR is added into that, then PTP's loading is done. Then there is they give a fluorescence actually whenever there's signal. So mainly there is a sulfurase DNA capture beads are there which contains millions of copies of single clonally amplified fragments. So here this one. Um, then let's say if it is starting from this side, we have our polymerase and it's annealed there with the PPI phosphatase 1 and with the APS added. Sulfurase and luciferase they got attached to it, giving rise to ATP, and then the light is there with oxyluciferin. That's the basic with the light that is coming from there. It's a little bit of principle for it. And about Illumina sequencing, so we start from here maybe. So you have your DNA, then you add a prepared genomic DNA sample with the help of adapters here. So adapters are attached on the right hand side. Then these adapters are attached to a surface which has DNA to it. Then bind single stranded fragments randomly to the inside surface of the flow of cell channels. So you have your adapters here, then you have a DNA fragments, and then dense law of primers on downside. So afterwards, what happened? Bridge amplification is happened. So we have added unlabeled nucleotides on from the top, yeah, and then they will start to make a complementary DNA from there by adding a nucleotides on the top there. So then they will be attached to it. So there will be something denaturing the double bond standard molecules. So they will stay there up. Then first chemical cycle determine the first base. So whatever the cycles they are producing on the top, they will initiate the first sequencing cycle and add four labeled reversible terminators, primers, and DNA polymerase enzymes to flow cell. Then with the help of laser, you see different sequencing like GTCA, CTAG, TTAC, GGAC, like that. So image of first chemistry cycle could be seen, and then we can make a sequence out of it like GCTGA. Okay. So this is how Illumina Solexa workflow works like. And this is Abby's solid workflow. This is not so important one actually, so we will can skip that. Then there's iron torrent. About all these principles, I had one virtual video for the same. Let's continue.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is a gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below.
Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The iron proton machine is used mostly in clinical settings because it is able to provide a sufficient size output.
So here you can see here that the data that they're showing is with the pyrosequencing sequence by synthesis, by ligation and ion semiconductor, how how the data looks like. So with the pyrosequencing, it seems only the rhabdopsis and the bacteria we can see, but whereas in the sequence by synthesis, it seems quite good. It's okay, but with the sequence of ligation, it's much better than with here. It's ion semiconductor. It's quite even better. So you can see how all difference are working out today.
Within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com. And from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service. Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
So that was about the summary of all the. Oops. Okay, that was a summary of all about next generation sequencing, including Sanger also. So I hope this is clear for today. Uh, so what are the applications? So these are the various applications. Please go through them. Genomes, resequencing of human exons. So I will not read them. You can please go through this. Then studying the function of gene protein, some basic principles and nomenclature. So what you can see is here from the mutant organisms, with the help of genetic analysis of screening of DNA library, we produce cloned genes, and with the DNA sequencing, we produce proteins, which are localized. Biochemical studies could be done. Determination of structures are done. Further, this could go back to do the database search to identify protein coding sequencing and PCR isolation of corresponding genes or gene inactivation. So in vivo, in living organisms, we can do the study of a function of gene or proteins using bacteria, yeast, monkey, or Christmas tree also. In ex vivo, we can do it with the organ explant, example tissue biopsy, and in vitro we can use reagent tube, synthetic setup, and in silico is a computer-based simulation or database analysis. So remember, because we are sometimes really confused with what is in vivo, what is ex vivo, what is in vitro, what is in silico. So remember these terms, quite important. Yeah, in vivo is with the living organisms like bacteria, yeast. Ex vivo is in organ explants like tissue biopsy in a cancer cell tissue. In vitro is the reagent tube in the synthetic setup, like in the Appendorf or in the, yeah, your reagent tube. In silico is done with the computer-based simulation or data analysis. So, what are the advantages of culture set over the intact organisms? So, of course, working with the whole organism, like whole rat or whole guinea pigs, whole sheep. Somebody in Ireland, he has a farm of sheeps. What he does is, he's a famous scientist also. So as a professor, he when he wanted to check the ischemic cardiomyopathy, so he was creating uh, some problems to the sheeps, to their heart, and then checking the various echocardiographs, various others cardiomyopathic readouts. So he was using his own sheeps from the farm for the experiments. So which is are quite good for the professor, but it's somehow very difficult to take their lives. I am against actually. So in my case, when I was working my PhD, I was cutting around 60 rats every Tuesday in the morning time, at 7 o'clock. So I was cutting their head, forehead, then decapitating his chest out of it, then bringing out the heart out of them, and then we are doing the primary isolation of the heart cells, cardiomyocytes. So I did that for like almost 2 years every morning. I feel horrible to kill rats by your own hands, seeing the blood coming out, push, like that sounds comes also, and it's white. It's all organs are in your hands suddenly, and they are newly born. They are just 3 days born. That's why neonatal rats they are called as. So they are newly born, and yeah, it's horrible experience. Sorry to share that. So you can, but this is how you can. So you don't have to with the humans. You can't do that. It will be really strange if somebody is doing so, and it will be very unethical to do so. So that's why the mice rats could be sacrificed to help us actually. to find out the answers that we are looking for. I will show you also my research part, what I did during my PhD, which has a very latest technology, including bioinformatics, proteomics, mass spectrometry, cloning, IP. We also did cell culture. A lot of cell culture was there. So I will show you all results also, how research is done, how first the question is prepared, how the first hypothesis is made, then theory is made, then whatever the research has been done, that you have to write it down. Then your lab books plays a big role in that. Your protocols that you're writing, your networking matters a lot, how many scientists you meet in every day-to-day life. So these all matters while experiencing the PhD. So let's come to the point now. So why we use cells over the whole intact organisms? They are more homogeneous cells than the tissues. They can control experimental conditions. We can isolate single cells to grow in the colony of genetically homogeneous clone cells, and they are ethically non-controversial. Yeah. So we can use it like bacteria, yeast, vertebrates. In plants, we can use Arabidopsis, Thaliana. In vertebrates, we can use mouse, human, rat, hamsters, all these parts. In USA, I was for one conference research. It's called Gordon Research Conference. If somebody is in their PhD, please try to go for Gordon Research Conference. It's called GRCs of your own field. It's quite expensive though, but if you can arrange to go there, you can meet really top-notch Nobel Prize winner scientists. I got this chance to have dinner with two Nobel Prize winners. One was the Japs, and I don't remember their names, and one was from America. So they their personality was itself was like very um, yeah. What shall I say? I have no words for them. So they were very humble and being so touched with the students and helping each other in their research very openly, giving us our answers to our any questions about career or anything. And yeah, so it's a dream come true conference. So try to look for conferences, guys. If any of you is in their masters or in your PhD, to go for a broad conference or within India, they are really helpful, very helpful for your projects. So growth of microorganisms in culture like E. coli and yeast colonies, like you can see bacterial colonies to be grown here and yeast colonies grown here. So you can have to study various gene functions. You can use these also for your experiments. So this is shown you one small experiments how replica plating is done. So you have a rich medium master plate with different bacterial colonies. So you take some sterile velvet covered stamp. You took it and put in the two different medias. One is with lacking arginine, one is having the arginine. So in this case, we have the missing colonies which has arginine mutant, which requires arginine. So it's required arginine to grow, but doesn't have arginine, so that clone will not grow. Others will grow. But in this case, we have arginine mutant colony growing in the minimal media because it has arginine in it. So this is, you can check it like that. Then growth of animal cell culture. So here you can see requires rich media including essential amino acids, vitamins, salts, glucose, and serum. Most grow on the special solid surface, like blood cells grow in suspension. So this is single cell mouse cell. This is colony of human cells, and this is many colonies in the petri dish. So yeah. So this is the replication from this we also did actually. So this is how the human cells and mouse cells growth pattern looks like. So growth or rate of culture. So it goes first exponentially, then it is stable in the phase 2, then it starts to decrease like this in cell senescence. Then the cell senescence happens, that is cell is not anymore working in that case. In the mouse cell, first it starts to initially lose the growth potential, then there's a senescence for this time in this case, then start to have emergence from the immortal variant cell lines. So this is a 30-day culture, and then it's after 60-day culture, and this was for the 50 cell generations in that case. So we have three phase here. We also have a kind of three phase here too, in the mouse cells and the human cells respectively. These are the fibroblasts that are cultured, you can see, and differentiated afterwards. Very famous one, NIH 3T3. If you got chance to work in any foreign labs, so you will see using day and night. Yeah, if you there's in New York, there's a CSTR, Cold Harbor Spring Lab, Cold Harbor Spring Labs. Yeah, so there is like if you're working there, you have to work day and night. Actually, you hardly get time to eat, and you will be using a lot of experiments, lot of things you will be doing, and you are just talking about science each day when you are starting your research. So I have seen that kind of life. Once I was part of it, but if you want to continue, it's all exciting. You have to work for your papers, you have to fight for your research, you have to fight for your science, you have to fight for your ideas. So that's how that's what I'm telling you, some internal future, that's how the somehow sometimes science really works. It looks good outside, but inside is also good, but you have to be very hard working and also a smart scientist to do so, to go to achieve your dreams that you're looking for. Okay, on that note, I will take a 5 minutes break here. Then we will continue with this. Just need to get my energy back and then see you at 21:05 then. Yeah. See you soon. You also take some break. Have something. So welcome back, welcome back. Um, I hope you all refresh. Okay, these are the fibroblast. Let's continue. I want to finish with this today for sure, and I want to bring you to the next part of yeah, a little bit about DNA fingerprinting. Um, so these are undifferentiated cells. So if you see into more details, so cells are like open up, like they are quite like somebody have pushed them, yeah. But when they get differentiated, they make the structure like how it should be. So this is the DAPI, I guess, not DAPI, the red dye, uh, red fluorescent dye. So you can see the structures like this in them. So they are more in differentiated form. So now let's about the, this is quite, I have to say, important part. Sorry, transgenic mice and knockout mice. So that's the most of the research has been done. So any group that you will join in future, whether in the Cold Spring Harbor lab or in Max Planck or our CSIR labs in India, or any ICER Institute, IISc, Bangalore Indian Institute of Science, or any IITs group, wherever the biology is going on top. So most of the people in the molecular biology, they are having knockout mouse or transgenic mouse or some conditional mouse or knock-in mouse. So they have these mouse models where you can check for functioning of something. So in my case, when I was doing my PhD, so I was looking for function of cysteines. Yeah, so cysteines are meant to be do disulfide bridge. So if the cysteine in the protein is here and cysteine is there, and you have your alpha helix structure, and the other side is there also, and then these cysteines make disulfide bridges together as a protein. So what in the normal way, this is your normal structure, but what if in the mouse you removed one cysteine with serine? You remove this and you add serine to that, and that is known as knock in. So you bring remove one thing and you bring it new one here. So because of that, these disulfide bridges will be broken and the protein structure will be changed and the protein function will be changed. Structure and function, both are changed. So you can check then what happened in the control state mice and the disease state mice. What if we give the disease in the mice and the control state mice with the uh, with this knock in and knock in part, the one which is having a knock in and a ki we say. So you can check what happened in their case. So same you can check with the knockdown in a gene, like if there is gene 1, gene 2, gene 3, 2, 3. So you remove this gene. Yeah, let's join them. These are your introns. So once you remove these genes, then you check the functioning, because the function of that gene is removed, and then check whether the disease that we are looking, the, like in our case when we were working, so we were looking at the myocardial infarction, MI. So whether that has been reduced or it has been increased after removing that gene. So you can check many aspects, not only in the heart but also in the liver, in the lung, in the eyes, in the brain, in the stomach. You can check any aspect by removing or addition of gene. So that's the basic part of our transgenic mice, in which we add gene, there's a gain of function, and we remove the gene, there's a loss of function. Or we can partial knockdown with the help of siRNAs, like I did in my master thesis. I showed you with the metastasis cells, where we HUVEX human umbilical endothelial cells are vascular endothelial cells are uh, yeah, siRNA knocked down by them. So these are the various. Also, you can do conditional and tissue specific knockdown like that way also. So that was some general idea about it. Some definitions. What is genotype? You know, which makes the whole genetic makeup. Phenotype makes the functions and physical appearance of it, and whereas there could be a mutation could happen in the wild type, and that could lead to the different genotype, and hence you can see a different phenotype. So organisms that develop such mutations are called as mutants, and mutations may be part of genotype or also called as mutations in them. So they are also called haploids and diploids. Haploids are the one which has single copy of each chromosomes, and phenotype is a sequence of that one copy, and diploids are the two copies of each chromosomes, and thus two copies of gene. So diploids that carry identical alleles are called homozygous, which have different alleles are called heterozygous. This is just to let you know the normal to revise your genetics part. So here you can see at the bottom, wild type, genotype is this, phenotype looks wild types. There is one wild type and one normal. Then the mutant, there is a dominant gene present, and then and then there is a normal, other allele is normal, and you have mutants. If both are present, then it's a mutant also. But one is recessive, other is also remain empty, then it's a wild type again. Yeah, but both are recessive, then also it's a mutant. So this is how some phenotypes looks like. This is just one example, nothing to be to think about why is it dominant is mutant, why is recessive. So nothing to think about that. So these are various genotype linked diseases are written, like sickle cell anemia. This comes quite common in the questions from the autosomal recessive and autosomal dominant. So what are autosomal recessive diseases? What are the various examples of autosomal dominant? So you should remember these names: Huntington disease, hypercholesterolemia, X-linked recessive genes are the DMD, hemophilia A, so phenylketonuria, Tay-Sachs disease. So yeah, please go through them. So this is shown you some crossovers, how crossovers could let the affected and non-affected, how the carrier and uncarrier is being generated. So in case of autosomal dominant Huntington disease, what is happening is, uh, this is your female, this is your male, or this is your male, sorry, this is your female part. So here A is HD, Huntington disease is there, which is affected when they are cross. Male and females are produced, so the one on the left with the HD genes will be affected, with the one with not will be not affected. Same with the autosomal recessive. When it is recessive, like cystic fibrosis, so whenever, if the CFTR gene if present in both cases, only then it will be affected, otherwise not. So these are carriers. This is also carriers, mother and father. But in their kids, only the affected one will be the first one with the both genes having present CFTR. So only then it will be affected. And something with the X-linked recessive genes like DMD, Duchenne muscular dystrophy, in which there is a male X plus Y, and the female is a carrier in this case. And then you cross over the male ones here. You can see are affected with the DMD genes, but the carrier ones are not. The females are not, because it's X dominant. Yeah. So because it was in the males. That's why it was in the males. That's why this one is affected, and the carriers are not, because it's a male dominant one. So these are the various examples of mutation in Drosophila, which is in genetics kind of base model, where mutations may cause some subtle changes or may produce significant changes in the development of cellular function and appearance of behavior. So this is, you can see a red eye in the normal case, but in the mutant the colors has been removed. This is normal two wing, and this is four wing in the UBX mutant. If you remove this UBX gene, you can see these four wings things in that case. So, so what is gene knockout? Is the means of inactivation of gene, whereas transgene means addition of gene to that, additional gene to that. So we can check further about this. So this is a method to show you how knockout mutation is done. How embryonic, first of all, you have to produce embryonic stem cells to generate a knockout mutation, and then from them, these embryonic stem cells are put in up into the female embryo, and then from the blastocele, first in the blastocele, then the female embryo, then it goes for further. Yeah, production, that what you're looking for. You have to make a lot of crossovers and make things done. So this is basically shown you a method of how it is done. So here you can see that you have neo-R gene and TKHCV gene here. Gene X is replaced with the construct. So gene X that we want to, it has been removed from there. Then there is a two steps, either homologous recombination, non-homologous recombination. In the homologous recombination, ESL DNA has GNAX, GN targeted, and the mutation in the gene X resistance to the G14 and Ganiclover. But in this case, you have no mutation in gene X and cellular resistance to sensitive cells. So at the end, by this method, you produce your ES cells with targeted disruption in the gene X. So whatever the genes that you don't want into your things, so they are removed at the end of it. So these genes are then added to the blastocele of the cavity of embryo, then surgically transferred to the pseudo-pregnant female. Then you have a chiremic having a both stripes and brown color here, and you cross it with the black one. So while crossing, what you see is you produce three generations: one with without stripes, two with without stripes, one with the black again, and at the end, by these methods, you have a knockout mouse. But this seems quite. I mean, this is quite easily shown, but when it's done, comes to the two practically, it takes around 2 years or sometimes 3 years to produce these mices actually. Yeah, it took around 2 to 3 years to produce these mices, but there are this Charles Rivers company, they are doing this professionally. They can generate you within few months also, 2 or 3 months, these mices. But some labs that I was working with was the lab of Professor Offermans. He's a highly renowned scientist in the world. So they were producing these knockout mice by themselves with the GPCRs uh, and G protein coupled receptors, yeah, GPCRs, and they are working with lot of different mice and different level of diseases. Actually, they didn't let any other disease left to be unopened. They were working with the cardiac field. They were working with the liver diseases. They were working with the skeletal muscle diseases. So they have touched all the part of diseases in the field of research. So that's the beauty of creating knockout mouse. Once you create a mouse, then you can check all the function of the other organs also, of the heart, of the liver, of the lungs, of the yeah circulatory system, endocrine system, how enzyme system is working, how the breathing rate is going on with the mice, how much level of extension they can live actually. So you can check all these different parameters after creating your knockout mouse. This is some another way of creating a knockout mouse by Cree method, which you can disrupt your second like from here second part is removed from there. So guys, this is not so necessary to teach you in details, okay? Just have a look over it, how it is done. But to be sure to make it more clear for you, this is to create a transgenic mice with the mice with have a growth hormone. You can have a large mice than the normal one. So like this you can create a transgenic mice with the growth hormones with the normal one, so like elephantasis or something like that. uh, to say that I will now bring to you main part. Actually, that's what I was running for, the latest technology that in 2016 one of my colleague was doing by not telling anyone. So we were keep asking what research you are doing, but she never tell us. But when she was successful, she told us that she was working with the CRISPR-Cas9. So CRISPR-Cas9 is a technology which is quite recently quite hot. Instead of creating your knockout mouse, knock-in mouse, you can do everything in the cell or in the mouse by using these CRISPR-Cas9 models, and it's quite fast to do so, and its results are quite good and fast. And most of the IITs actually they are creating a lot of workshop for this. Just look for IIT Bombay or I don't know, IIT Delhi might be. They are creating a lot of CRISPR-Cas9 workshops, and you can think for maybe joining them. So in our when we were teaching, so they came for workshop in our case. So I will show you a little bit about CRISPR-Cas9 now. So go for it. You some of might you know this technique, some of you might not. So this is for everyone. Then enjoy this CRISPR-Cas9.
Gene knockout is a common technique in studying gene function. In this practice, the gene of interest is disrupted by altering the cell's genomic DNA. Any phenotype that results from gene knockout gives insight into the function of the disabled gene. Conventional knockout experiments are frequently achieved in animals, but need a huge investment of time, money, and labor. Therefore, these experiments are usually restricted to well-funded labs or core facilities. This has presented a large hurdle for researchers exploring gene function. The breakthrough of CRISPR technology has turned the tide and made gene knockout a much more accessible tool for many more research labs. The key revolution brought about by CRISPR is the creation of double-stranded breaks sequenced specifically. With CRISPR, scientists can create double-stranded breaks at virtually any site in the genome. To repair the double-strand breaks generated by CRISPR, there are two main mechanisms: non-homologous end joining and homology-directed repair, also known as homologous recombination. With non-homologous end joining, the ends of the double-strand breaks are simply reconnected together, and this process is highly error-prone, introducing mutations, insertions, and deletions, and in many cases resulting in gene knockout. Homology-directed repair, on the other hand, utilizes a repair template, where desired changes are flanked by left and right homologous sequences. Through homologous recombination, the desired changes are incorporated into the genome. The CRISPR knockout knock-in kits utilizes the homology-directed repair system. To plan an HDR-backed knockout experiment using plasmids, the researcher needs to create the following constructs. 1. A plasmid coding for the gRNA, targeting the intended site for double-stranded cleavage. 2. A plasmid coding for the Cas9 protein. And 3. A plasmid serving as the repair template, with a designed sequence flanked by the homologous arm region of the cleavage site, or donor vector. Origin has created pre-designed kits to make knockout experiments even easier. For each gene locus, we have created a specific kit. The first two elements are provided in a single all-in-one vector. The gRNA vector, targeting the start of the coding sequence, was cloned into a vector containing the expression cassette for Cas9 protein. The donor vector was also created with a knock-in GFP puromycin cassette, flanked by the homologous arms of the chosen cleavage site. This permits the selection of knockout cells via puromycin marker. The promoterless GFP provides additional utility to monitor any upstream promoter activity that might be present. To start your knockout experiment, simply co-transfect a gRNA vector with a donor DNA plasmid into the cells. The Cas9 enzyme will cut a double-stranded break near the start codon, and through homologous recombination, the functional cassette will be integrated into the genome, replacing target gene. In the end, 3 events are achieved. 1. The target gene is knocked out through gene disruption. 2. GFP will be driven by the endogenous gene promoter, providing added information of promoter strength. And 3. The puromycin marker under the PGK promoter is integrated into the genome, enabling the selection of successfully edited cells. The protocol starts with co-transfection. There will be 3 separate experiments. One of the 2 gRNA vectors co-transfected with the donor vector, scramble control with donor vector. After transfection, the cells must be passaged for about 20 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin.
Sorry to interrupt. So that's the main principle here written for you, that you have this PCAS guide plasmid with the Cas9 CMV motor and gRNA. So they already made this, origin company made them by themselves, and then you have this the gene that has been knock out or knock in has been removed with the GFP, and there's a puramycin being added also in that way. So it's like CRE locks, Cree locks method that you have in the knockout mouse, but it has a Cas9 way of doing it. So that's something new and fast, and you can see the results quite instantly. So I will continue.
Autosomal form of donor vector is lost. This diagram before genomic control with donor vector. After transfection, the cells must be passaged for about 20 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin resistance. Culturing the transfected cells for 20 days ensures that the episomal form of donor vector is lost. This diagram shows the details of transfected cell passaging prior to puromycin application and selection. For HEK 392 cells, 48 hours post-transfection is considered G1 cells, at which point the cells are split 1 to 10 and grown for 3 more days. At G2, CRISPR-targeted cleavage and gene editing should be finished, but the percentage of edited cells will be small. You can perform optional genomic DNA analysis using G2 cells. If the data are negative, you can redo the analysis after puromycin selection. After G7, apply puromycin selection using the doses appropriate for your cells. To summarize the CRISPR knockout knock-in protocol described so far, Step 1. Co-transfect one of the gRNA vectors with the donor vector, along with the appropriate controls. Step 2. Passage transfected cells for around 20 days. Step 3. Apply puromycin selection and isolate individual cell colonies. Please note that the drug selection doses need to be determined by kill curve for each cell line. 5 days after puromycin selection of transfected HEK 293 cells, almost all cells in the 2 control transfections died. However, as expected, the co-transfection with the gRNA construct produced puromycin-resistant cells. The 4th step is to analyze the puromycin-positive cells to detect the knockout. If you have a good protein-specific antibody, you can use Western blotting to evaluate the knockout. It is best to use this method after isolating single colonies. Another method is genomic PCR to verify the correct GFP puromycin integration in the genome, followed by sequencing to confirm the integration. To amplify only the edited allele, design PCR primers following these rules. The forward primer should be upstream of the 5' end of the left homologous arm of the donor vector, and the reverse primer should be in the GFP region. In this example, knockout ATG5 gene was conducted by the protocol. Genomic PCR products of the correct size were amplified from the gRNA constructs, plus the donor co-transfected cells, and not from the scramble control transfected cells. The PCR products were further sequenced using the PCR primers to confirm the correct integration. Sequencing data using the forward primer shows the correct integration of the 5' end of the left homologous arm in the donor vector. The reverse sequencing primer confirmed the correct integration of GFP puromycin cassette. The original ATG5 gene was replaced with GFP. Origene offers genome-wide human and mouse CRISPR knockout knock-in kits. With these complete, ready-to-use kits can knock out almost any gene from the genome and knock in the GFP and puromycin cassette simultaneously. You can study the consequences of the gene disruption and potentially do a promoter study at the same time. Origene also has other functional cassettes to provide different fluorescent proteins, luciferase, or different selection markers. If you wish to have the CRISPR knockout kit created a different donor vector, please visit our website to request a custom project. To learn more, please visit the Origene website and our YouTube channel.
So that's it. A simple method for the knockout with the CRISPR-Cas9. As you can see, within 20 days, within your cells, you can see the effects that you're looking for, and make the desired results for your experiments. Just a second. So I guess we are done. Oh, there's. Would you like to see another one? I have created one for the knock-in actually, but I think that's enough. Would you like to go for? Why chatting is not coming? Is something got stopped or what? All is good? All awake? Okay, okay, great, great. But yeah, for Biocon, if somebody is interested to work with Biocon, there is a thing called Biocon Academy. That's a window to work in Biocon. Very few people knows about this, Biocon Academy. So there you have to do some courses, and then after done with the course with them, they will allow you to be part of the Biocon family. So that's one. Just go through this Biocon Academy, go through the courses. You can find it what is best for you, and then you can be part of Biocon company. So virtual lab, I can look for maybe tomorrow. I can write it down. I can write it down. Any other questions? So which that you demand for? Please let me know. I can look for CRISPR-Cas9 a virtual lab if there is any, because things get easy to understand then. That's always. Yeah, it's Biocon is a big company. Majumdar Shaw, who started with papain with papaya enzyme, his company in her garage. Now a million dollar companies. She's amazing. She's a role model actually for me. Uh, yeah. Okay. Next generation sequencing. I will write that if there's any virtual lab for that. Okay. So I can. There is one more video. I think it will be easy. I will show you that. If we are into the video mode today, let's continue the video mode then. Oops, what is this? Ah, yeah, here it is. This is to create a knock in mouse. Knock in. Knock out.
The ability to insert or knock in a gene at a specific locus is a tremendously useful. The ability to insert or knock in a gene at a specific locus is a tremendously useful method for dissecting disease mechanisms. However, in previous years, the insertion of a foreign gene has been far too tedious and expensive for many labs to perform. Thanks to the recent development of CRISPR-Cas9 technology, gene knock-in experiments have now become a very popular technique in modern molecular biology. In contrast to the random integration events by the lentiviral system, CRISPR utilizes a guide RNA that directs gene insertion with extraordinary specificity. In particular, the AAV-S1 safe harbor site is often a preferred target site for gene knock-ins. This is because it enables consistent and robust transgene expression with minimal toxic effects. At this safe harbor site, the surrounding DNA maintains an open conformation, enabling stable expression of the newly inserted transgene. This makes safe harbor sites, such as the AAV-S1 locus on human chromosome 19 and the ROSA26 locus on mouse chromosome 6, very popular sites for targeted knock-in experiments. In this case study, we will walk you through how a red fluorescent protein gene was knocked into human embryonic kidney cells at the AAV-S1 safe harbor site. We will also demonstrate how the knock-in was verified using a PCR-based method. To begin, an sgRNA was designed to target the human AAV-S1 locus. Software analysis was performed to ensure maximum efficacy of on-target cleavage while avoiding any off-target effects. The resulting guide sequence was used to construct ABM's AAV-S1 targeting all-in-one vector. This vector expresses the Cas9 endonuclease under a CMV promoter and the sgRNA under a U6 promoter. To achieve the knock-in, a donor plasmid containing the gene of interest is required to provide a template for homology-directed repair. The HDR pathway is a mechanism in cells that repairs double-stranded breaks by using a DNA repair template homologous to the sites flanking upstream and downstream of the break site. To enable HDR-mediated gene insertion, the gene of interest must be flanked by such homology arms. Therefore, a second plasmid was constructed by cloning an RFP pyromycin expression cassette into ABM's repair template plasmid for the AAV-S1 locus. The resulting AAV-S1 donor RFP gene under a CMV promoter and a pyromycin's resistance marker under a PGK promoter. Once both plasmids were ready, they were introduced into HEC293 cells using ABM's transfection reagent, DNA fectin. Following transfection, the cells were passaged 10 times to dilute out the episomal donor vector. Following, the resulting HEC293 cells were given 3 to 4 weeks to grow in the presence of pyromycin to select for the cells with successful knock-in of the RFP pyromycin resistance cassette. Cells transfected with both AAV-S1 targeting all-in-one vector and the AAV-S1 donor RFP plasmid were healthy after 4 weeks, and greater than 95% of HEC293 cells were successfully expressing RFP. Control cells that were not transfected with the vectors died after pyromycin treatment. PCR genotyping was carried out to confirm that RFP was successfully knocked in at the AAV-S1 site. In this method, 2 primers were designed. Primer 1, targeting the 5' homology arm upstream of RFP, and Primer 2, targeting a site within the RFP pyromycin resistance cassette. Gene knock-in was confirmed when a PCR product of 1.1 kb was observed, indicating that primers 1 and 2 were both able to bind to the edited genome to enable successful PCR amplification. In the case of a non-successful knock-in or in the presence of wild-type control cells, no PCR products were observed, as primer 2 could not anneal to the target site and enable amplification. We hope this case study has demonstrated how the CRISPR-based gene knock-in technique can make your exploration of gene function amazingly easy and efficient. CRISPR-Cas9-mediated gene knock-in allows for biallelic modifications of some genes to be generated with ease, with phenotypic changes in mice observable in a single generation, an unprecedented time frame. Let ABM's team of CRISPR scientists help you knock in any gene into any cell line you want, all with minimal toxic effects at the AAV-S1 or ROSA26 safe harbor sites. Explore ABM's extensive portfolio of customizable CRISPR services and products today, and start knocking in genes, tags, markers, and more. To make it even easier for you, simply describe your desired knock-in, and we'll take care of the rest.
Oh, that was the last one. So they used a totally different system. They totally different system using these two plasmids they made. This one and that one, and the donor plasmid, target gene, and this one. So there was one part they brought two. Yeah, this one, this Cascade 9 AAVS and AAVS1. So I had different strategies. So each company has their own strategies, and they're selling them to the market to do so. So this is also in science. Marketing is there. Marketing is in everywhere. So we are done finally with our day 3, day 4. Actually, it was together. Day 3 and day 4. They were together. Uh, so next part, let's continue. I will not take much time for you today. Uh, let's go for DNA fingerprinting. So, DNA fingerprinting is done with the help of your restriction enzymes to check between two possible crime people who did that, whether the person is not or was it different one. You can check it with the help of DNA fingerprinting. Uh, so why use DNA fingerprinting? It tells whether that the specimens that we are looking is of the individual. Sequences are variable and can therefore be used for identifying characteristics. It has advantages over the source of evidence like fingerprints, blood type, etc. It is highly accurate and can be gathered from trace crime scene evidence. So one way is by doing RFLP. These are various markers of yours, like restriction fragment length, polymorphism, uh, and then we will deal with all markers also: RFLP, RAPD, AFLP, SNTRs, SSRs, molecular markers. All these parts will be deal also by tomorrow, I guess. Restriction enzymes are molecules that can cut DNA into pieces. Each enzyme cuts a very specific DNA sequence. While all human beings share roughly the same DNA sequence, but they are a little bit different. Because of that, we see change in the gel. So I want to show you this RFLP animation. So here you have restriction enzyme BAM HI, and you have CGATCC. So you have restriction enzyme came, it cut it like that. Then you have your sticky ends like we discussed before. So let's say in the individual one, it can cut at 4 different sites. So A, B, C, D. So you have 5 fragments after cutting from the 4 fragments, obviously. And individual 2, it can cut the same restriction enzyme at the 3 sites actually. So you have four different parts of DNA. So individual has one has 5, individual 2 has 4 different parts. So that's how essentially DNA has been cut into enzymes, and we have a DNA fragments of various sizes. So each individual having a different sizes. Restriction enzymes cut each person's at different points, and fragment of different sizes will travel at different distances along a gel when an electric current is passed through it. So you can see different banding will be there for different person. Like for individual one, 2 closed bands are there. For individual two, a separate band is there with different sizes. Another different. So if individual one or in the crime scene spots blood samples that we have collected, if they both matches each other, then we say that okay, that's the culprit. So that's how you can find it out with the RFLP technique. So a little bit about this introduction, how it could help to define the law and science. So forensic science can be defined as the intersection of law and science. So there are also in India, if somebody is interested to do go for forensic studies also, you can do from masters in forensic science, where you will learn a lot of techniques, what's happening at the crime sites to check, take the blood sample, checking, doing DNA isolation, then doing a PCR, then checking via gel electrophoresis. So all these things are possible in the forensic science field. So first photography fingerprint was done in 1985. That is in DNA fingerprinting. In 1985 it was done. So DNA fragments show unique patterns from one person to the next. So on basis on that paternity, we use to have forensic evidence. So specimens could be licked envelope, dirty laundry, a cigarette butt, or saliva. So we can use any part of our sample, and special precaution has to be taken while doing so. Wear your gloves. Disposable instruments has to be there. Avoid talking and sneezing. Avoid touching samples with your skin. So you have seen that when they are into the crime site, how they take the samples very carefully. But in our Sushant case, it seems totally opposite of that. And it's very sad to bring that point for us today. That with what happened with the Mumbai police, and I don't know. I'm not here to judge anyone, but it seems that's why CBI came play to the role. So everyone, so we say in India, Satyamev Jayate. So truth should be win in that case also. So whosoever was culprit, whether he's suicide or whether it was a murdered, so we should have the truth to be win in that case. Uh, so yeah, so enemies for the evidence are sunlight. So sometimes they can remove the way the yeah, high temperature, bacteria, moisture. They can remove the effect that it was before. Ideal sample is 1 ml of fresh whole blood, treated with EDTA. That's the ideal sample if you want to test anything with somebody. So then further, RFLP, so we have discussed this already with the RFLP, how individual 1 and 2. So like an individual 1, it cut it from here in this part, and restriction enzymes. So like GAATTC, so it's cut it at the G and also here also. And whereas in individual 2, it cuts only from one side, not from the that side. So you have here three different fragments, A, B, C, and whereas we have only D and C fragments there in the individual two. Yeah. Uh, so RFE fragments can be separated by gel electrophoresis. So those fragments that you did, so individual one will show like A, C, and B with different sizes, and in the individual two, it will show like C and D. Yeah, like this. So that's how RFLP works. Like RFLP main work goes like this. And further molecular technique where DNA is transferred onto membrane from agarose gel and probe is hybridized. That is your DNA. We did this quite number of times. Southern blotting, yeah. So I will touch this part later. Then there is a variable number tandem repeats. So sequence are repeated multiple times and number of repeats varies from person to persons. So like in one person it's 5 repeats, like GATC, 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 they are being repeated. In some person it's 2 times it's repeats. So these are called your variable number tandem repeats. So they mostly occurs in the introns and can be amplified by PCR and agarose gels to produce unique DNA fingerprints. So like in this case, you have 5 repeats here on this side, and this is a 2 repeats on this side. PCR, we did this quite number of times. Lab technique used to amplify segments of DNA. So this is also could be used in our DNA fingerprinting to amplify the DNA that we are looking for. So we know we need what we need. We need DNA to amplify the specific portion that we are looking for. We need primers, we need DNA polymerase, and we need nucleotides. So reactions are placed in the machine, thermocycler, then it goes to different temperatures. First it go 94, then it's go to 50, then it's go to 72, then DNA polymerase elongate to the newly strands, and then you have this cycle for 30 times, then you have your PCR generated DNA sequence molecule. So that's how it is. So, what are the applications? It could help in diagnosing the diseases. Various diseases could be checked out with that. So, like if, this is normal, this is diseased, and this is the carrier one, and this is how it's yeah, level of degree you can check. And this is a pedigree analysis has been done, where your parents then siblings goes like that, and you can check in different patterns how your in gel electrophoresis with the DNA fragments you can check in different things. So like in this case, if the parents have all three bands, so the children's has like two here, and here is all three in this case, and then one in that case. It could help in the paternity testing. It could help in the forensic, like we discussed. Uh, so whosoever the victim, crime scene, and suspect, we can check in the three different patterns, various um, yeah amino acids which are playing role. So in genealogy animations, thank you for today. I think that's it for today. I will not go further. So this I will share with you today. And also I have this, oops, sorry. Uh, so I have discussed with you about restriction enzymes quite number of time. Uh, so this is only 20 slides thingy. Maybe we do this tomorrow by open mind, fresh mind, because I don't want to go too much on that. It's just tell us about how the nomenclature of restriction enzymes are done, where there are various types of restriction enzymes, how DNA ligase, kinase, receptor transcriptase, clinofragments works, how the mode of action is done, star activity. So this will be more about restriction enzymes. For tomorrow. So maybe I do it tomorrow then with a fresh mind. Because fast things will be not good for us. And okay, so that's it, I guess, for today. So your questions, I'm open to your questions now. If I know, I will tell them today. Otherwise, I can tell you tomorrow also about questions. So tomorrow is weekend. So enjoy your weekend, guys. Mostly due to lockdown, every day is a weekend actually. Sorry to say that, but now things are as opening. Uh, so enjoy your Saturday, Sunday. Yeah, IISER Pune. I was there once in IISER Pune. It's quite famous for various and next generation sequencing is being done there at very good way, and also if somebody is interested with the structure biology, with the X-ray crystallography, they had really good facilities and very famous scientists also. I have seen the faculty level and how the administration level, library, everything, it's really up to the mark for the international level. So ICEs are also playing really good running competition to the international level, I have to admit. Yes, bache. So any questions, students? So in meantime, please subscribe to the channel also. Uh, let's see how this YouTube thing is works. So this is my first time I'm on online platform. So while using YouTube, they say we can do a lot of things actually uh, while playing. So I wanna bring new ideas to for you. Uh, so that uh, we can bring new things for you. Yeah. So I want to show you something. I just forgot. Mm, yeah, there is a virtual lab of cloning. We do that tomorrow also. Just remember me that we have to do virtual cloning. I will write down in my agenda uh, cloning for tomorrow. So no worries about your reviews. If you need some time, I know it's very less time from now onwards, maybe 5 or 6 days. Uh, so I will share also today presentation you have already, I guess. Yeah, I will share the rest of the videos also. Uh, and today, DNA fingerprinting, a technique that has been used between both to catch the culprit using RFLP. So where it was. COVID will stay with us, I think, for this year. COVID will stay here with us for 2020 at least. So we have to live with it. We have to wear mask mostly and clean our hands when we come back from outside work. Maybe try to change your clothes if possible. And don't be afraid of it. It's become our family now, COVID. And if even though if it happened to us, we will recover out of it. Don't worry. Just increase your immunity. Have exercise daily. Drink a lot of warm water. Eat like avoid fast food as much as possible, and try to eat boiled vegetables, which is not possible at your age actually. I can understand, but try to increase your immunity as much as possible. That's only we can do. Avoid junk food. Wake up in the morning, do some yoga with this pranayam and om vilom. So this increases our lung capacity to have good air within us. And yeah, how to revise for today's. Yeah, it's that's why I was telling you each day everything very normal things for you, but today I want to show you something extraordinary, like which is happening in the world, and this is future. Like CRISPR is future, NGS is future, uh, DNA fingerprinting is already going on. So all in all, things are happening. So I had already shared these files with you. Go through them and try to look for some references books. Try to do search today for the same bioinformatics. But the thing is, I was more into the proteomics and mass spectrometry and little bit of molecular biology. So I'm not a bioinformatician. uh, so I'm reading nowadays actually bioinformatics by myself at home. So I have one book by Methods and Applications by Rastogi. So bioinformatics methods and application by Rastogi. So we are also, guys, one more thing I want to tell. I have a one friend from Denmark. She did from Sweden, Lund University, her master's in bioinformatics. So she will be taking care of your a 10-day workshop in bioinformatics. So earlier they were asking me to do so, but I told them that I'm not good enough for bioinformatics. So uh, yeah, so we will try to have that workshop for you, and also I'm trying to have one proteomics workshop. Of that, I'm quite looking forward to it actually. After the biochemistry and MBBT, we are thinking for a third one is proteomics and bioinformatics. These two topics we will be touching with 10 days workshop with 30 hours classes. So this is the next month deal coming up. So already the content is ready, and for bioinformatics there will be she's at the moment health advisory at the Denmark company as a bioinformatician, and she's it will be good for you also guys to go for that course. Uh, yeah, proteomics. I can show you a little bit about proteomics classes also in this later part, because proteomics is quite hot. With the mass spectrometer you can do a lot of techniques, lot of things to identify. Okay, on that note, I will say good night to everyone. Thank you for your cooperation. Thank you for being such a great audience. And I humbly thanks each one of you who stayed till now for with us. And now have your dinner and take care, sleep well, good night, stay safe. And thank you very much. I'm really humble that you're giving such appreciation. Thank you and good night. Take care. Bye bye. So I will end the stream. Good night.